everybody, and we're going to be continuing with the genetics lecture in here today, and then in laboratory we're going to do more genetics problems. So please bring your genetics lecture handout to laboratory, because again I'm going to be using part of the laboratory period to cover some of the genetics topics. In this coming Friday, in here you'll take your usual lecture quiz in here, and it will carry, walk around. It will ask questions about the mitosis chapter, some mitosis questions, but it will also ask some questions about the genetics chapter that we're on now. So be prepared to answer questions from both of those. All right, well, let's jump back in. So up there on the screen is Gregor Mendel, and he's the one, remember, who discovered these laws of genetics. And just to recap some major things here, he said for every gene there was always two alleles, one dominant and one recessive, and he used the capital letter for the dominant allele and the lowercase for the recessive allele. But he said since each of us have two of each gene, because we're diploid, we get a set of chromosomes from mom and a set of chromosomes from dad, he said each of us will have two of each gene, and so the possible genotypes, the possible combinations of alleles that a person can have or an organism can have, could be two of the dominant allele, or one dominant and one recessive allele, or two of the recessive alleles. And these were homozygous dominant, heterozygous, and homozygous recessive. Now, his concept of the dominant allele was that it controls the phenotype, the physical characteristic, whenever it's present. And the recessive allele only controls the phenotype if there's no dominant allele present. And so if we want to think of this in terms of pea plants, like the ones Mendel was using, the dominant allele for color made for purple pea plants. Like that. And so notice it doesn't matter whether you have two of the dominant allele or one of the dominant allele, you get the same color purple. What's going on here, remember, is that this is an enzyme that manufactures purple pigment. And just like having two car keys or one car key doesn't make your car run any better. All you need is one copy of that enzyme, and it will make enough of the purple pigment to make the plant purple. But the recessive alleles are usually non-functional versions of the gene, a non-functional version of that enzyme, so it doesn't make any purple pigment, and you get a white-colored pea like that. And we call this the dominant phenotype, and this is the recessive phenotype. All right, so mid-1800s, Mendel worked out those laws of genetics, and that's the basic concepts that he worked out. What they found since Mendel's time is that his laws of genetics apply to almost all of the genes that have been analyzed and discovered, but there are some exceptions to the rule. Some genes don't exactly follow his rules of genetics, and we call those genes and the patterns that they follow non-Mendelian genetics, patterns of inheritance that don't follow Mendel's rules. And there are several types of non-Mendelian genetics. There's one called codominance, one called epistasis, incomplete dominance, continuous variation, linked genes, and sex-linked traits. And what I want to do is go down this list and explain in what ways the patterns of inheritance of these differ from Mendelian genetics and why they differ from Mendelian genetics. And as kind of a preview, I'm going to skip epistasis and linked genes. Those are interesting concepts, but they're a little bit beyond what we have time for in this class. But Hartnell does offer a genetics class now, and if you like genetics, I think it's kind of a cool subject. You might take it there and you'll get a complete list then. Anyway, let's talk about codominance for a second. When Mendel was formulating his laws of genetics, he said there's only one dominant allele. Well, he said there's only two alleles for each gene, one dominant and one recessive. In other words, there's only one dominant allele. What they found for some genes is that there can be more than one dominant allele. There still can be a recessive allele. That hasn't changed, but there's more than one dominant allele. And usually that just means this. Remember, dominant alleles are usually ones that are encoding for a functional enzyme, but there's two different versions of that functional enzyme. So you can get two different phenotypes from the two different dominant alleles. And the classic example of that, of codominance, is something called blood type. Here we go. Imagine this shape right here is one of your blood cells. There's an enzyme that can add molecules to the surface of your blood cells called antigen molecules, and they call that the I enzyme. I'm not sure exactly why they picked I, but it's enzyme I. And what this enzyme does is grab some molecules called antigens and stick them on the surface of your blood cells. Antigens. Now, why it's important for you to have these antigens on your blood cells, or I should say why this enzyme thinks it's important that you should have some antigens on your blood cells, I'm not exactly sure, but that's what this enzyme does. And so as long as you have a functional copy of this, as long as you have the big I allele, you'll have some antigens on your red blood cells. But there's a recessive allele also, which, of course, Mendel would have said we need to represent with little I, that is a non-functioning version of that enzyme. And so it's not able to put any antigens on your red blood cells. So far so good. It sounds like good old normal Mendelian genetics. But here's the interesting part. There's more than one dominant allele. There's more than one version of capital I. There's one that's called capital IA, and it puts a certain antigen on there called the A antigen. And there's a different dominant version of that dominant allele, capital IB, that puts the B antigen, a different antigen, on your red blood cells. Well, you may wonder, what exactly are these antigens? They're actually short polysaccharides. And again, why our blood cells can have those little sugars on them, I'm not exactly sure, but it's just part of our physiology. Okay, so if there's these two dominant alleles, the capital IA and capital IB, you can make a chart of the possible genotypes and phenotypes that can be out there. If a person has genotype capital IA, or capital I little a, then they have a functional copy of that enzyme that makes the A antigen, so they're going to have the A antigen on their blood cells. Oh yeah, and that's called blood type A, if you have just the A antigen on your cells. If your genotype is capital IB, or capital IB little I, then you have functional copy or functional copies of the dominant allele that makes the B antigen, so you're blood type B. Here's the interesting part. If you have both of those, capital I and capital IB, you have both the functional enzymes, and so look what happens to your blood cells. They have the A antigen and the B antigen simultaneously. And if you're little I, you don't have any functional copy of the enzyme, and so no antigens get added to your cells, and so you're said to be blood type O. So those are the phenotypes. The phenotypes are the four blood types, AB, A, B, and O. But there's 6 possible genotypes. Big IA, big IB. Well, you can see them for yourself there. All right, and maybe I should get back up here. Notice that this meets the definition of codominance. There's more than one dominant allele, and organisms that have both dominant alleles will fully show both traits. And that's what we're seeing here. Both the A antigen and the B antigen are simultaneously present, so they're fully showing both traits. That is codominance. All right, I'm going to have you guys do some Punnett square questions now. See if you got this. Let's say, for the sake of example, that there's a woman who is blood type O. And there's a gentleman who is blood type A. And so using a Punnett square, is it in any way possible that they can have an O-type child? Who says yes? Who says no? Who doesn't have an opinion? I guess the rest of you. Well, it comes out that it is possible, and let's look at it with a Punnett square. All right, so there's the woman. Do we know her genotype for sure? Yeah, if she's blood type O, that's the recessive phenotype. And anytime someone shows the recessive phenotype, you know their genotype. She's got to be little I, little I. And so when she makes her eggs, in meiosis, each egg only gets one of her two genes, but it doesn't really matter which one. So all her eggs are going to be carrying the little I allele for blood. Do we know his genotype? Well, yeah, we don't know for sure is the point there. We know he's blood type A, but he might be that, or he might be that. He might be homozygous dominant for capital IA, or he might be heterozygous with capital IA, little a. But it is possible that he is this. This would also give him blood type A. So it is possible that that's what he is. And if you fill in the Punnett square, look what it shows. Every kid they have has a 50% chance of being blood type O, and every kid they have has a 50% chance of being blood type A. So yes, it is possible for a blood type O woman and a blood type A man to have blood type O kids. All right, here's one I'm going to let you guys solve on your own. There's a woman who's blood type B, and she has a kid who's blood type O. And there's somebody who she thinks is the father who's blood type AB. Is it possible that he's the dad? Well, you guys are good. Yes, I guess you didn't even have to draw the Punnett square. But yes, he could not possibly be the father. Oops. Oh, actually, I guess I got ahead of myself here, but I'll do the Punnett square there. So we know she is B. Let's put her over to the left. Do we know whether she's capital IB, or capital IB lowercase I? She has to be lowercase I because her child has to be little I, and so the kid must have gotten a little I from both parents, so she's got to have a little I. So we know she is this. Well, okay, that's what she is. So half of her eggs will have the capital IB allele, and half of her eggs will have the little I allele, the recessive allele. And the man who might be the father is blood type AB, so he's got to be that. And if you do the Punnett square, there's no way that these two people could together make a type O baby. They can have a bunch of other kids. They have blood type AB kids, or blood type B kids, or blood type A kids, or blood type B kids, but no blood type O kids. Alrighty. Here's another example of codominance. There is this disease, sickle cell anemia, which I know I've mentioned a few times before. it relates to the hemoglobin protein. The hemoglobin protein is inside red blood cells, and its job is to carry oxygen. And it's actually made up of 4 polypeptide chains. Remember, some proteins are made out of more than one polypeptide. And one of the polypeptide chains is 146 amino acids long, and this shows you the first 8 or so of those amino acids. The gene that encodes that particular protein is called the HB allele, or that's the HB allele of it, and they put uppercase A because in a sense it's a dominant allele for the normal protein. There's another allele which is codominant called HBS for the sickle cell version of that, and the difference is there's a small change in the nucleotide sequence, which leads to 1 amino acid out of the 146 being changed, but it causes the protein to fold into a different way, and because the proteins are folded in a different way, the blood cells end up with a strange shape, usually kind of a half-moon sickle shape. That's where the name of the disease comes from. So people who have this allele tend to have sickle-shaped blood cells. And there's some other effects also, other than just the shape of the blood cells. It causes a certain amount of weakness, and so there's definitely some health